Good afternoon, everyone. Welcome back. Today we're going to talk about emerging viruses. Building on last time, we talked about evolution. So what is an emerging virus? Here's the definition. A causative agent of a new or previously unrecognized infection. So it could be a brand new virus that we've never seen before, or it can be a virus that we've known about, but suddenly causes a different disease. Either one we call an emerging virus. Now, this term was coined in the 1990s, but of course, viruses have been emerging forever. It's just that we began to recognize them. And the crossover, today we're going to be concerned about the crossover of viruses from non-human animals into humans. It can go back the other way. It can go from humans into animals. And among animals, they can spread viruses, but we're mostly going to talk about from animals to people. And this begins around 11,000 years ago when we're no longer hunter-gatherers. We're not living in bands of 50 to 100 people that are going out and hunting for food. We're starting to farm and raise animals. And so now we can live in greater and greater numbers. And the animals that we contact then start to invade us. Plus, the populations get big enough to sustain outbreaks of viruses. So, agriculture and commerce, that's all the beginning of today's story. And only really since the 90s have become good at detecting new viruses. It's hard to do, as you will see. So emerging viruses can have an expanded host range with a disease that was not previously obvious. An example is Zika virus was known since 1947. It never caused microcephaly until 2015. Suddenly in Brazil, babies were born with microcephaly, small heads, as a consequence of Zika virus infection. We'd never seen it before. So that's an emerging virus, even though we knew Zika virus for many years. So the name for transmission of a virus from a wild or domesticated animal to a human is a zoonosis. And sometimes we talk about reverse zoonoses, where humans give them back to animals. And sometimes these events, so I'll sometimes refer to them as spillover events from the non-human animal to the human. Sometimes it establishes a new virus in humans. It becomes a human virus. So the movement of SIV, simian immunodeficiency virus, from chimps to humans established a new viral disease, AIDS. The movement of SARS-CoV-2 from bats to humans established a new disease, COVID. But there are many times when the virus spillover is not sustainable. So Ebolavirus from bats to humans, it doesn't become a human virus. Eventually the infections stop, and the next outbreak is a new spillover from animals. MERS coronavirus is the same way. Every little outbreak of MERS coronavirus disease is a spillover from camels. Even West Nile virus, every human case is a spillover from a bird through a mosquito to humans, because humans don't have enough viremia for mosquitoes to be able to bite them and take enough virus to transmit to someone else, right? So you have to distinguish these two major classes of zoonoses, the ones that become a human disease. Then we don't call it a zoonosis anymore. SARS-CoV-2 is no longer a zoonosis. It's the virus formerly known as a zoonosis. And at least one person got that inside joke. So when it becomes a human virus, we don't call it a zoonosis anymore. It has its origins in a zoonosis, but Ebolavirus, MERS, West Nile, we call them zoonoses still. All right, so here are some interesting origins of the human pathogens that we know about today. And these are number of genera of viruses. So we have families like Picornaviridae, and then within the Picornaviridae would be the enteroviruses. That's the genus. And within enterovirus, you'd have poliovirus and Coxsackie and rhinovirus, et cetera, which would be the species. So this is the number of genera and where they came from. So 31 viruses belonging to 31 genera, the orange bar, those are human viruses today. They did come from an animal. Every human virus came from some animal somewhere. There's no question about it. And so 31 that we have are established human pathogens like SARS-CoV-2, HIV-1, measles, polio, et cetera, that crossed over a long time ago. 37 are zoonotic viruses. Every outbreak comes from an animal. So many bat viruses are in this category. We'll talk about some of them today. 16 came from our hominid ancestors, the precursors to the Homo species, which you may have learned about at some point. They're not called Homo, they're like Australopithecus, for example. We acquired viruses from them, 16 genera. And then finally, we acquired viruses in 6 genera from our Homo siblings, like Homo neanderthalensis and denisovans and whatever else might be out there. So that's where these, and in the case of the Australopithecine passage and the Neanderthals and the Denisovans, those ultimately came from animals as well. So they don't always come directly into humans, but they can be indirect as well. So that's the origin of many of the viruses that we see today. So what drives this spillover of viruses from animals into humans? There are a lot of factors, and the biggest one is human population growth. I mean, look at this. This is years versus billions of people. It's just amazing how much incredible population growth has occurred in the planet. And the consequence is people are crowded together in certain places, and we occupy every corner of the planet. We invade environments where animals once were alone, and we get their viruses. This is a major factor, but there are many other factors as well, and they're shown here. Travel, obviously, is a big one. That's a snapshot from this website where you can track every flight in the air at any given time. You can click on one and see what the flight number is and so forth. Look at that. I mean, it's interesting that some countries are excluded from substantial travel, but for the most part, this can spread pathogens globally. And then all the factors on the left here. Dams. Making a dam changes the environment and changes the animals and therefore the viruses we can get from them. Irrigation, deforestation, we're going to see an example of that because it pushes animals out of their habitat into human habitats. What else should we talk? Long distance transport of livestock and birds. We talked about how pigs are shipped around last time. They carry influenza viruses with them. Of course, blood transfusions can spread diseases. That's why we check the blood supply, but before we did it for many viruses, that's how we spread those infections. Transplanting organs from other animals. It's not yet clear if this is going to be an issue, but we're worried about it. A lot of people get, say, heart valves and even whole hearts from pigs. Pigs have their own viruses. They have endogenous retroviruses. They're called porcine endogenous retroviruses, PERVs. Okay, and those, I once asked a guy on a podcast, have you heard of PERVs? Not the kind that you're talking about. But these are retroviruses integrated into the genome of the pig. There's 60 of them. And so we're worried that if you put a pig heart in a human, they're going to start to replicate and cause problems. So the lab has actually made knockouts of all of those PERVs using CRISPR. And now we are beginning to use organs from those pigs. Let's see, air travel we mentioned, urbanization, daycare centers, which didn't exist in my day when I was a kid. Now you put lots of kids together, they share their viruses, they bring them home, hot tubs, because they can be improperly sanitized. Same thing with air conditioning, those on the top of all these buildings, those water towers, they're great places for microbes to grow. Used tires we talked about, transporting mosquitoes. Climate change. Yes, climate change is real, and as temperatures increase, it affects the animals. Mosquitoes' ranges change. The Aedes aegypti, which is a tropical mosquito, is moving farther and farther north. And it's going to take its diseases with it. And as animals, if it gets too hot for a certain animal in a habitat, it's going to move and it's going to take its viruses with them. So this is no question changing the landscape of disease ecology. Here's an example of how many viruses are out there. This is the Amazon North region of Brazil. And each of those lines, which you can't read, is viruses that we know are there in different parts of the Amazon North. 183 known arthropod-borne viruses and other vertebrate viruses. And some of them have been isolated. But that's all that we know about. There are lots in there because there's a huge fauna, right, in the Amazon. And the problem is that we're cutting it down. So people are going in and cutting it down, and they're contacting the viruses, and that's the source of a new infection. So there are all kinds of factors that lead to spillovers, and today we're going to talk through some examples of them. But some of them are summarized here, where we have the virus family, and then the factors leading to the emergence. So for example, a dengue virus, urban population density, open water storage favoring mosquito breeding like used tires. Ebola, contact with the host, which most likely bats, but we're not sure. Hantan virus, contact with rodents during the rice harvest in places like Korea. And we'll talk about Hendra today, fruit bat transmission. We've talked a little bit about influenza virus, its reservoir in aquatic birds. We'll talk about HIV next time. Bushmeat trade is a big one. That's when you hunt animals in the forest like bats or non-human primates, and you eat them and you get infected in the process. MERS, camel husbandry, again, Nipah, fruit bats. SARS-1 and SARS-2, open air livestock markets, etc. So many factors, and for each virus that emerges, there's usually a specific factor that is leading to its emergence. Now, don't forget what we learned last time. There's a huge role for evolution in emergence of new viruses. Why? Because in every animal in nature, whether it's a bat or a pig or a duck or whatever, each one is infected with many viruses, and they exist as a quasi-species, right? A collection of mutants. And every replication cycle, the population diversifies. And just at random, in some bat somewhere, there may be a virus that can infect humans. It's random. There's no thought process involved. And needless to say, as I've told you before, it's random mutation. And so because there's so many bats, for example, in the world, and they have so many viruses, eventually just at random, one is going to emerge that can infect people. And then the key is, of course, that bat has to find a person or contact a person. And so the diversity of viruses in these animals leads to the ability to move to new hosts and environments. And that's part of the quasi-species. Within the quasi-species is a virus that can reproduce in another host, whether it's another non-human animal or a human. So this is a key part of understanding how this works. And the diversity in nature is huge. Just think of this in light of this, lab origin, nonsensical lab origin idea for SARS-CoV-2. The idea that a person could make a virus that would do this is absurd because in nature, it's so rare that it happens. You have to go through so many genome iterations, and then you have to have contact. It just doesn't make any sense because the people who propose it don't think about this properly. All right, so now before we talk about specific examples of spillovers, I want to talk about different categories of interactions between hosts and viruses. It's kind of a framework so that you understand what's happening in natural hosts and spillover hosts. So we have 4 categories that we put viruses into, depending on how they interact with their host. We have the stable host-virus relationship, they're in green. So that's just like it sounds. It's a system that maintains the virus, so it could be humans and poliovirus or humans and measles virus. It's a virus that's been in that host for a long period of time. Then we have an evolving host-virus relationship. This involves passage of virus from a stable relationship to a different host. It could be a different species or the same species. We'll see examples of both today. And that, you can see here, the big red arrow shows that Viruses can go from stable host-virus relationships into evolving ones simply by changing hosts. That's a major pathway there. And we have a dead-end host-virus relationship, one-way passage to different species. So again, it comes from a stable host-virus relationship. A bat that has a stable virus associated with it passes the virus on to another species. It reproduces and never spreads. It's a dead end. And then there's the resistant host, where there's no infection whatsoever. And this happens all the time, of course. Viruses get introduced into different hosts, and they fail to even multiply. And so they're of no consequence, but they certainly happen. All right, so let's look at these in some detail. So in a stable host-virus relationship, which the majority of infections in the world fall into this category, so both the host and the virus multiply, right? Both survive because otherwise this wouldn't exist. Some of them are permanent. So humans are the only host for measles virus. They're the only host for herpes simplex virus, human cytomegalovirus, smallpox virus, and many others. All of these viruses came originally from a non-human host and changed sufficiently in humans so that now it's a human virus and it doesn't reproduce in any other host. So that's an example of a stable, permanent host-virus relationship. And as far as we can tell, it will go on forever until maybe we can, well, if humans die out as a species, then the viruses are gone too, obviously, but we may be able to eradicate some of these viruses, measles virus perhaps. And in some cases, you have infection of more than one species. So a stable relationship doesn't have to be just one kind of host. It can be multiple hosts. And so influenza viruses can be in various animal hosts and in humans. Flaviviruses like dengue virus can be in humans and non-human primates as well as mosquitoes, and toga viruses as well. So that's the stable host-virus relationship. Then the evolving one, that involves passage of virus from a stable interaction to a new host. And it's called evolving because it's difficult to predict what's going to happen. When the virus goes into a new host, it often changes extensively as it adapts to the new host. And that's that with dead end, or what we call emerging infections. So what is the outcome of this? It could be benign, or it could be death. And if it's death or something slightly less, then it brings itself to our attention and we work on it. So what are some examples of this movement of virus from a stable to an evolving host-virus relationship? Well, as Old World colonists came to the US from Europe, they brought their viruses with them. Smallpox and measles viruses emerged in the Middle East, spread to Europe, and then came to the Americas by the colonists. They didn't exist in the Americas. And so they were introduced into a completely naive population, and a lot of death resulted, as we'll see in a moment. West Nile virus was never in the Western Hemisphere before 1999. It was present elsewhere. It was brought over into New York City in 99. It spread throughout New York, caused some disease, and the virus evolved as it's spreading. It's an evolving host-virus relationship. And then our artificial introduction of myxoma virus into the Australian rabbits to eliminate them is another example of a movement from a stable host-virus relationship. Myxoma had its own host somewhere into a new host. And you can see how it evolved. In the first year, 99% lethality, and then less in the subsequent years. So that's an evolving host-virus relationship. Now, the dead end is another example of what can happen when a virus goes from a stable host-virus relationship into a host where there's not a lot of transmission. It's not very effective. So this is another frequent outcome of cross-species infection. That's why the red arrow is a major pathway as it is to evolving and dead end. The key here is there's no sustained transmission from the new infected host to others of the same species. And an example is Ebolavirus, which comes from an animal reservoir in the forest into chimps, into gorillas, into humans. Not really sustained transmission. Now, you may say, but what about the 2015 outbreak in West Africa, 25,000 cases? Yeah, the problem there was for 3 months, they didn't know it was Ebola virus, and they were taking care of patients in hospitals, and they were transmitting it through patient care, droplets, and close contact and so forth. As soon as we imposed infection control, the transmission stopped. It didn't become a human virus that could transmit indefinitely. So even though this sounds confusing, I call it a dead end. It doesn't become a human virus. That's the point there. It can spread if people are taking care of each other. And it's such a serious infection that people had to be hospitalized, and then that was where a lot of the transmission occurred. Also, when people died, their family, the practice in these countries is that the family prepares the body for burial, and then the family gets infected by the close contact. H5N1 virus, avian influenza H5N1, another example of a dead end. It comes into people from birds, and that's it. It never transmits to anyone else. Now, everyone's worried about it because it's appearing in cows and goats and all kinds of animals in the US. It's in birds globally, and they're worried that it could adapt, it could change so that a random virus would infect humans, but it hasn't done that in 50 years. I don't know why, but now that there are more animals infected, it's concerning, of course. These kinds of relationships contribute very little to the spread of the natural infection. There have been 800 human infections with H5N1. That does nothing for the maintenance of the virus on Earth. What maintains the virus is the virus infecting birds, and that maintains it big time. So these little dead-end infections, while they could be lethal and serious for us, they contribute nothing to the spread of the natural infection in the natural host. So here's some examples of stable and dead-end host-virus relationships. So this is a virus that is transmitted by mosquitoes, and the host for this virus is wild birds, and you can have wild birds of different species here. And so a mosquito will bite a bird and pick up virus. The bird is already infected. The mosquito picks up virus, spreads it to another bird. So there's a cycle where the mosquito spreads the virus from bird to bird. So the natural host is the bird. The virus reproduces to high levels in the bird, and when the mosquito takes a blood meal, there's enough virus in the blood to transmit to another bird. And this can be complicated. It can involve other birds like chickens on farms can be bitten by these mosquitoes, and they can be infected and pass on the virus to others. And other mosquito species can also feed on the same bird and transmit the virus to other hosts other than birds. So it could be a complicated natural cycle. Now, occasionally mosquitoes bite other species than birds and chickens. They can bite horses or humans, and they can give them severe disease, but the humans do not transmit it to anyone else. The humans are dead-end hosts. Why is that? They don't make enough virus in the blood so that a mosquito taking a blood meal from a human would be able to transmit it to another host. You need a certain amount of virus, and because this is a brand new virus spilling over into people, it just doesn't work. And West Nile is the same way. West Nile, in fact, is a natural reservoirs, birds and other rodents. And when they bite horses and people, it's a dead-end infection. So every infected horse, every infected human is a spillover from a mosquito via another mammal. Okay, so both stable and dead-end host virus relationships are shown on this slide. Here's another one that just involves ticks. The same idea. Here's a virus spread among rodents by ticks. Ticks feed on rodents, of course, as well as many other mammals. And they can take blood meals and pick up viruses and spread it to other rodents. Then the ticks can, of course, bite other animals if they have contact with them, like cows can get infected. And then it's been shown for some of these viruses that the virus is in cow's milk and can spread to people. It's a good way, it's a good reason to pasteurize your milk. You know, a lot of people now are into unpasteurized milk. You gotta be crazy. I'm sorry, folks who really like it, but Pasteur did a good thing by pasteurizing milk. People were dying, and now you don't want to pasteurize it to have a natural experience? Well, you're gonna get viruses, and you're gonna get bacteria as well. So that's here if this is not pasteurized, because the pasteurization process will inactivate viruses, including avian H5N1. The ticks can also bite people. And again, they're dead-end hosts because you're very unlikely to get bitten by another tick, but even if you were, there's not enough virus in your blood to transmit it. But many infections happen this way, and many new infections. It was a disease a number of years ago, first seen in the US, where farmers were developing high fever and thrombocytopenia. Turned out to be a tick-borne virus that we'd never seen before. And the farmers are out in the fields all day, so they get bitten by ticks, right? They'd come home with 10 ticks all over them, take them off and not think anything of it, but it turned out that they were transmitting a new virus to them. So an emerging infection where a virus, the kind where a virus is now coming from a non-human animal into a human, not the one where, like Zika, it's a new manifestation. There are two steps, I think in general we can think about it. So you have the viruses multiplying in some animals and generating their quasi-species. And among those quasi-species, there's maybe one or a few that can reproduce in humans. And why, we don't often know. Receptor change, permissivity changes. We just don't know for the most part because it's very hard to get these original hosts for most spillover viruses. So then these animals have to encounter a human in some way. And as I said, we are providing lots of ways for these encounters nowadays. But beyond that, the virus has to become established in that first person and then disseminate. So let's say there's a farmer in the middle of nowhere who gets infected with a bat virus, but he doesn't go anywhere. He has no family. He doesn't go anywhere. If he lives, that's the end of the infection, right? But if he has a family, maybe he could infect the family, and then the family goes to a market and infects other people. So you need to have a chain of events, right? So these spillovers probably happen all the time, but most of them fail because there isn't the encounter of the right population of people. And as I said, I think this happens all the time. I think in all kinds of ecological situations, there are multiple spillovers of viruses from animals into humans, and we just never detect them because the virus isn't right for reproducing in humans. Our immune system takes care of it. I mean, even here in New York City, there's a lot of wildlife in Central Park. There are lots of raccoons, there's lots of squirrels and rodents, of course, and their excrement is there, and very few infections originate here in a very populous city, so it's just not the right collection of viruses. So a lot of these happen, we don't know about it, and they're not transmitted. Sometimes that farmer gets infected, but the farmer doesn't go to meet anyone else, doesn't reproduce to high enough levels. A lot of conditions have to be met in order for these infections to take off. So I'm impressing that on you because it's not trivial. These things are very rare. And when we do see one like SARS-CoV-2, it involved an extraordinary series of events that is very rare and can only happen in nature. Now, if a virus is going to make it out of the animal into humans, it has to find susceptible cells, right, where it can bind to the receptors, and they have to be permissive. And the virus has to be able to reproduce to high levels in those cells, evade immunity, and then spread within the organism. And then beyond that initial human population, density is really important. You have to have a lot of people together, as I told you. And that's why, after the establishment of commerce and agriculture, that's when we really begin to see emerging infections in humanity. And the health of the population is important. If it's a generally healthy population, it will be harder for viruses to establish themselves. And the key here again is that for a new virus to establish itself, you have to have serial infections. You have to have a chain of infections from the first person infected. And that's why the great illustration is the open air market in Wuhan. Wuhan is a transportation center. Millions of people, 10 to 12 million people live there, and people move in and out every day. So here you have an open air meat market where you bring animals from the countryside with their viruses. They contact people. It's a perfect scenario for that kind of emergence. And as you'll see, it's not the first time that it happened. It's at least the second time. All right, so let's look at some spillovers and learn some principles from them. So first, smallpox virus, a scourge of humanity that killed many tens of millions of people over the years, and we have eradicated, actually, by vaccination, amazingly. When we look at different smallpox genomes and sequences them and look at related smallpox viruses and their hosts, it looks, by that analysis, that this virus emerged 3,000 to 4,000 years ago in East Africa. And here's East Africa here, this little red circle. And we think it was transmitted from camels to gerbils. So camels originated in the Americas, and then they spread into South America, and they spread into Asia through land bridges that were present back then. And the camels end up in many places in India and also in the Middle East. And so we think the camels came here about 3,000 to 4,000 years ago, and they encountered gerbils, which have an overlapping range. That's the hatched oval there. And that's a gerbil up there on the upper right. People often have them as pets. And there's evidence that gerbil pox viruses are part of this story. So the virus went from camels to gerbils as a consequence of the camels moving, right? The new host encounter. These were introduced to Africa 3,500 to 4,000 years ago. And sorry, I got it a little backwards. The virus was existing in gerbils. And when the camels came and contacted the gerbils, they picked up the smallpox-related virus and infected them. And then we got it from camels 3,000 to 4,000 years ago. So it's an interesting situation where new animal contacts is a part of the catalyst here. So that's smallpox virus. Measles virus is a little simpler story, but measles virus is very related to rinderpest virus, which is a pathogen of cows. And we think measles evolved from rinderpest virus when humans first began to domesticate cattle in the 11th and 12th centuries. So remember, we were hunter-gatherers for many years. We had to hunt our food. And then we got smart and learned that some of those animals you could not only eat, but you could put them in fences and they would reproduce and you could have your meat there without having to chase it. And so that put us in contact with cows more frequently and with their viruses, and rinderpest virus is thought to have spilled over into humans about when we began to domesticate cattle. And you have to have a certain number of people for a measles outbreak to occur, between 250,000 and 500,000 people. So as human populations began to congregate in cities, that's when measles virus infections took off. And so this is thought to have, so domestication of cattle first happened in the Middle East, and then the virus emerged there and spread to Europe, and eventually to the Americas by colonization. And when it was introduced into the Americas, it destroyed many Native American populations because they had never seen this virus. They were completely naive, and they had no immunity. So it comes from a cow into people and then spreads around the world. And there are no airplanes. Remember, this just came over in boats from the colonists. Then we have diseases that we cause. I've just mentioned that in terms of measles. When Europeans decided to explore and see what was out there, they found the New World and they brought their viruses with them. And this is another example of how when you introduce a new virus into an immunologically naive population, it can have very fast spread and it can be quite lethal. And so, Cortez brought smallpox virus into the New World. And this is an illustration. You can see that individual there has smallpox lesions all over. And he had, a pitiful band of soldiers, and he was able to conquer this Aztec empire with millions of people because the virus he brought killed them, and they were unable to fight him. And similar conquests also happened because of bringing measles in to the New World as well. And so remember, smallpox originated in the Far East from camel to human spillover. It went to Europe and then came over to the New World from Europe. So those are examples of how changes in what humans do lead to emergence. And in fact, it's all about that. Everything is our fault, essentially. We screw everything up. We have cool inventions and arts and all that we make, but we screw the world up big time. So it's such a paradox. Humanity is an amazing paradox. But every virus spillover is our fault because of our interactions with nature. Yes. All right. So the question is, were they sick? Were they asymptomatic? There's some evidence that some of the sailors were sick. So you can get on the ship and be fine, and then it's such a long voyage that you get sick as you come over, right? There's some evidence that they actually purposely took blankets from those soldiers and gave it to the Native Americans to infect them. And then they're probably asymptomatically infected people as well. But they just had to mingle, and it spreads like crazy because there's no immunity in the population, right? And no face masks, none of that stuff, right? All right, so now let's talk about some other ways that viruses can emerge. And I love the example of poliovirus as an example of changes in populations, basically population immunity. So poliomyelitis has been around for thousands of years. We think this is an Egyptian carving from about 1500 BCE of a priest with a drop foot, which is typical of polio. So when you have paralysis of your leg muscles, of course they atrophy because you don't use them, and so you can see the atrophied muscle. But if you pick your leg up, so normally you can easily hold your foot up because your leg muscles hold it up. But he is paralyzed and his foot drops, and that's a classic sign of polio. We don't know if it was polio, but it looks like throughout history you can see examples of sporadic polio throughout history. But around 1900, the epidemiology changed and it became epidemic. You can see on the graph there, we're looking at annual cases versus the year. And if you go back way before 1905, all the way to 1500 BCE, about sporadic cases. But starting in the early 1900s, you get outbreaks. Look at thousands of cases until this one, 1916 here in the Northeast, a huge epidemic of polio, 28,000 cases. So what happened? The virus has always been around, right? So how did it change? So this is an emerging infection because now it's become epidemic. Its pattern of infection has changed. So that becomes an emerging virus. So what happened? Well, it turns out it's a disease of modern sanitation. Before 1900, polio circulated freely, infected everybody at birth. There were no toilets and sewers. You had chamber pots that we'd use in your house, and then you'd throw it out on the street, basically. The streets of New York were full of shit, human and horse shit. It was just horrible. But people didn't think this was a problem, right? Anyway, kids would get infected immediately after birth because viruses everywhere. Everybody's got poor hand hygiene. But you were protected from getting paralyzed because your mom had given you antibodies transplacentally, right? So you never got polio. It was very rare. Maybe the rare kid who got polio through the thousands of years was someone whose mom never got it and survived and didn't pass on the antibodies on. So the maternal antibodies prevent paralysis. But in 1900, you know what was invented? The toilet and sewage. Sewers, not sewage. Sewage always exists. Toilet and sewers, the greatest invention of humanity, which now puts all the feces away from the streets, and now kids don't get infected shortly after birth. They get infected later when they're 10 years old or 15 or 20 years old, and there are lots of them, and the virus somehow gets introduced because it's still around circulating, and boom, you have an epidemic of polio. So we call this a disease of modern sanitation. And I like to say, and that's why the improved sanitation leads to epidemic polio, changes the disease pattern, and it's an emerging infection. I like to say if we had genome sequencing in 1908, that would have sequenced the poliovirus from before 1900 and from after, they would have found some changes, right? Because when viruses reproduce, there are always changes. And they would have said, aha, these changes caused it to cause outbreaks, which would be wrong. And it's a nice example of how you can't rely on the genome sequence to tell you everything. So in these cases, you have to look further, and it's population immunity that's the issue. Another example of how changing climate and animal populations can lead to spillover. A new disease was noted in 1993 in the Four Corners area here of the US, where 4 states come together, called the Four Corners, and it was called hantavirus pulmonary syndrome. It's a severe pneumonia, a respiratory disease, causing fatality in very young people. It's never been seen before in the US, but it turns out it was present in other countries. Turns out the disease was caused by a new virus, which eventually got the name sin nombre, which you may know means no name, right? And the reason it says no name is because, well, the virus was first isolated in Muerto Canyon, which is a small village near the Four Corners, but the residents objected to calling a virus after their town. So the CDC tried a few different names. They didn't like any of them, so they finally called it sin nombre, and showing that they used to have a sense of humor, at least the CDC. So now this is a bunya virus. It's an RNA virus with 3 segments and an envelope. And the virus is endemic in the deer mouse, Paramiscus maniculatus. 30% of wild mice, these are Paramiscus species in particular, are positive for the virus. So what happened? Why didn't we see this before? Why in 92, 93, suddenly we saw an outbreak of this disease? Turns out there was a lot of rainfall in 92 and 93. And as a consequence, there were more piñon nuts that grow naturally in that area, and people like to eat them, but so do mice. And so the more piñon nuts that grew, the mouse population rose, and they went into people's houses more frequently than they had before. Now remember, 30% of Paramiscus mice are infected with this virus. They're fine. The mice are fine. It's a natural host for the virus. They have no problem with it. They excrete the virus in feces and in urine. And that, you know, if you see some mouse poop on the floor, what you do, you get a brush and you sweep it up. So you aerosolize the dust and you inhale it and you get hantavirus pulmonary syndrome. So humans are not the natural host. Every human infection is a spillover. It doesn't transmit among humans. It has not become a human virus. And then looking back after we isolated the virus, we found that there had been cases as early as 1959. So there it is. The mouse excrement has virus and you inhale it. So what do you do when you see mouse poop? You know, even here in New York, you could have mice harboring this virus. You shouldn't just sweep it up. You should spray it with a little bit of dilute bleach to inactivate what's in it and then pick it up later. And there have been cases of this in many states, as you can see here. Through 2021, hantapulmonary syndrome by state, 817 cases. And you can see a lot of them here out west, but we have some here. We have had 6 in New York State. Some of these are associated with campers upstate who camp on these platforms. They pitch their tent on this wooden platform, and the mice are underneath it. And so at night they come in and they deposit some infected material, and people can get infected that way. So the range of this mouse that harbors the virus, Paramiscus maniculatus, is shown here. So it's all throughout North America, as you can see. It's really just absent in the southeast US. But there are other mice that can harbor the same virus. Besides the Paramiscus is called the deer mouse. The white-footed mouse, the rice rat, and the cotton rat can all harbor the virus. So basically, really be careful if you're doing things with wild mice, okay? And the mice in your home are wild mice. They're not lab mice, right? So they can have this virus in them as well. So just be careful. So that's a great example of how climate change impacted the presence of a virus. Now, bats turn out to be a huge source of zoonotic infections, and we didn't know this until there were a couple of outbreaks caused by viruses from bats. And these include Nipah and Hendra viruses, which we will talk about. These viruses can cause severe disease in humans and domestic animals, and the bats seem to be fine with them. And many of them are found in fruit bats or flying foxes. So this is a flying fox here. You can see why it's called a flying fox, right? It looks a little bit like a fox. And the wingspan on these guys are like 3 to 4 feet. They're really big animals. And they harbor many of these viruses. In fact, this publication in 2011, bats host major mammalian paramyxoviruses. That's the family where measles is part of, measles virus. And Nipah and Hendra viruses were 2 viruses discovered, they're paramyxoviruses, to come from flying foxes and caused outbreaks. So let's take a look at this. Hendra virus was first discovered in Hendra, Australia in September 94. It's on the northeast coast of Australia there. And they didn't mind calling it the virus after their town of Hendra, so some people don't mind. There was an outbreak in a racehorse farm, and 14 racehorses were killed, and a trainer, and the trainers, of course, of racehorses are right near them. They're brushing them and taking care of them, so it's very easy for them to get infected. So one trainer actually died, and they investigated this disease, and it turned out to be a new virus, which they called Hendra viruses, which came from flying foxes, first to horses, and then from horses to humans. And so the idea is that the horses are in stables, and at night the bats fly in, and they excrete, they do their thing in the stable, and the urine has virus in it, and the horses get contaminated, they get infection, and then people can get infected. So this is a big problem because it's highly fatal, and racehorses are expensive, right? And so farmers, racehorse owners don't like this. So a vaccine has been developed. This is too rare in people to make a human vaccine, but you can vaccinate your horse, you protect your investment, and you protect people. So when we vaccinate wild animals, they're not wild, when we vaccinate non-human animals to protect humans, we call that a one world health approach. One world health means we're all in this together. We're not just humans worrying about ourselves. We have to worry about the other animals that are around us as well. One world health. Now, these bats have a wide range here. You can see not just the northern part of Australia, but all these other islands here and even up into the continent and Madagascar as well. So it's possible that they harbor these viruses in other places. But these infections continue to occur rarely, but they still occur in Australia. The other paramyxovirus that was discovered early on, 1998, is Nipah virus. This is the model virus for contagion, by the way. This was first identified in Malaysia in 1998. There was an outbreak of respiratory and neurological disease on pig farms in Malaysia. So here's Malaysia, right? There's Singapore down there. And the first outbreaks were in pig farms up here in the Perak district of Malaysia. So they noticed that the pigs were getting sick. They had respiratory disease. They had neurological problems. They identified this new virus, and 105 humans died because those are mainly the people taking care of the pigs. We're talking about a lot of pigs. This is a major protein source for people. So they actually culled 1 million pigs to stop this outbreak. And how do you kill 1 million pigs? Do you think you inject each one with a, no, you dig a big hole and you push them in it, unfortunately. It's really, you can find videos of this on YouTube. It's very sad. They're just, they're not all infected, but the only way you could stop this outbreak is to do that. So that stopped the outbreak. And the problem was the pig farms were outdoors. Pigs were just roaming on the grass or whatever, and the bats, they actually would plant mango trees in the pig orchards, and the mangoes would be food for the bats. The fruit bats would come at night, eat the mangoes, they would drop the bits on the ground, the pigs would eat it, and then they would get infected. So the fruit bats are harboring this virus. They excrete it in their urine. They're fine. They're the natural host. They infect pigs, and then the pigs infect humans. So this virus spread actually to Singapore because pigs were being shipped from Perak to Singapore. And when that was figured out, they stopped that. So there's no longer any more Nipah on Malaysia. But however, we now see Nipah infections in India and Bangladesh, which are transmitted in a different way by humans consuming date palm sap. So that's a palm tree there, and people like to tap it to drink the sap, and they put a pot in here to collect the sap overnight. And it turns out that the bats at night go in and drink it because they like it too, and then they pee in the pot and they put their virus in it. And so a very low-tech solution was to cover the collection pots, and that stops a lot of these outbreaks. There are currently still cases in parts of India and Bangladesh which are not associated with date palm collection, and we're not sure how those are happening. So why did this happen, right? Why did this happen in 1998? Well, there are a number of confounding events that were going on, and there's summarized here. First of all, that year there was an El Nino, which caused a drought in this region, and humans were doing, in Malaysia, they were cutting the forest down to make farms, and the forest is the habitat for the fruit bat. So the fruit bats didn't have anywhere to go. There was a lot of burning. They were burning the forest, so there was haze, and plus they were getting rid of the forest. That destroyed the wildlife habitat for the bat. They reduced their food supply. So they migrated. They migrated to these piggeries, these open piggeries in fruit orchards, and they contaminated the pigs, and that contaminated humans. So again, it's our fault. We're doing slash and burn, clearing, and destroying the fruit bat habitat. And you can see photographs online, you can search for it, of bats flying around in cities. Now, because they're forced out of their habitat. You can see in downtown city, a big bat flying around because it has nowhere else to go. So again, it's our fault that we did this. And so here's another summary of Nipah virus spillovers. So we have bats, flying foxes foraging on fruit trees. The pigs are exposed to the droppings. The pigs get sick, and the humans get sick. And then we have date palm sap harvesting. So people who are drinking date palm sap, which is not pasteurized, it's just drunk raw. The bats go in the pots and they drink it, contaminate the people, and the people get infected as well. And now we have people getting infected from unknown sources, maybe horses, and they're infected by a flyover event. We just don't know how some of these people are infected because there's no contact with date palm sap. But this is another example of how human activities lead to these spillovers. Another one that I really like is Argentine hemorrhagic fever, Junin virus. And this emerged after World War II. In Argentina, they wanted to make more corn fields to grow maize. So they were using herbicides to get rid of the native grasses in these areas, in the Pampa, right? The Pampa is a very rich area of Argentina with lots of interesting grasses. So they just sprayed them with herbicides to kill them all, and then they plant corn. So now corn is growing here, and a new shade-tolerant grass grows up. So that grass didn't grow before because it was competed out by the other grass, but now it's shady, and that grass can grow in the shade. So that grass begins to grow in the shadow of the corn. And it turns out that there's a new mouse that likes that grass, which comes in, the Calomus musculinus, which has a virus called Junin virus. And so the workers in the field who are harvesting the corn start to get hemorrhagic fever. And they described this fever first in 1953, and then the virus is isolated a couple of years later. So again, we're messing with the ecology. It changes the animals in the area, and we get their viruses. Ebolavirus is, of course, another example of a zoonosis. And we have had a number of outbreaks, mainly in Africa, starting in 1976. DRC was the first outbreak of Ebolavirus hemorrhagic fever, a very severe disease caused by a negative stranded filamentous virus enveloped called Ebolavirus. And over time, there have been a number of outbreaks. The diameter of these circles tells you how many cases are involved. Mostly there were a few hundred. Then suddenly, in 2015, there were over 25,000 cases in Western Africa where it hadn't been seen before. It was mostly limited to Central Africa. Now there was a new place. And as I said, it took them 3 months to respond, which allowed the virus to spread more extensively than it ever had before. But eventually, we were able to stop it by good infection controls. So what is going on here? So this is a classic zoonosis where someone gets infected by bushmeat trade. You go in the forest and you find animals that you can eat. It could be non-human primates or rodents or bats. And in many of these outbreaks, there's an index case that had contact with one of these animals. And then that infected person who's infected by, say, butchering the animal transmits it to other people. And these chains of infection are short because it requires really close contact with another person, like medical care involved or preparing a a body for burial. So here are two examples of outbreaks. The one in 1996 in Gabon, 37 cases. A chimp was found dead in the forest, and people found it and they started eating it for food. So you have to understand that it's very hard to find protein in many places in the world. So 18 people who were involved in butchering the animal became ill, and then they spread it to their family members, right? They bring the animal home and everyone eats it, they all get infected. Another one in Gabon, 60 cases. The index case was a hunter who lived in a forest camp. A dead chimp was found in the forest, was found to be infected with Ebolavirus. We don't know if that hunter actually had contact with it. So that's why in some cases there's a clear association with wildlife and in other cases not. The 2015 outbreak of Ebolaviruses began in Guinea in 2014. They did very extensive case tracing of this outbreak to find out what was going on. You can see here, the first case was a 2-year-old child who had onset of fever, black stool, and vomiting on December 2nd and died 4 days later. Then that child transmitted it to the sister here, who died also, the mother, the grandmother, and then a nurse and a village midwife, who then went another village and infected other people. And so that's how it ends up getting transmitted. The sister of S4, one of these cases here, attended a funeral and then spread it to other people at the funeral, and on and on and on. And we don't know how that child got its infection initially. We don't have any history of contact with wildlife, but presumably this virus comes from some wildlife source. So what is it? So these are called filoviruses, and there's another one that infects humans called Marburg virus, and that caused actually the first filovirus outbreak in people in Marburg, Germany in '76. And it's clear that that virus, its host is cave-dwelling fruit bats, because we've been able to isolate infectious virus from those bats. But for Ebola virus, we haven't been able to isolate infectious virus from any bat. We've got RNA, we've got antibodies in a couple of bats, but we never have isolated virus, and as a consequence, it's not really clear what the host is. But dead-end hosts are humans. Gorillas can be infected, they die. Chimpanzees are all dead-end hosts for this virus. And so we think bats are possibly reservoirs of Ebola viruses. They can spread it to chimps, to gorillas, to humans, and the chimps and gorillas can probably spread the virus to each other as well as to humans via the bushmeat trade. And there is some human-to-human transmission because you can get the virus by contact with infected blood or body fluids from someone who's sick and who's died. So in hospital or at home, this is easily to contaminate someone. Contaminated objects, it's not transmitted by insects, water, food, or aerosol. So it cannot transmit it to you just by breathing in the same room. It requires close contact. Let's now end with coronaviruses. So SARS-1 began in 2003. There used to be a service called ProMedMail that would archive interesting infectious disease events. And this was an email that was sent from this physician in February 10th. This morning I received this email and then searched your archives, found nothing. Does anyone know about this problem? Have you heard of an epidemic in Guangzhou? An acquaintance of mine from a teacher's chat room lives there and reports that the hospitals have been closed and people are dying. So in early February, there was an outbreak of severe respiratory disease in Guangzhou, but China didn't tell the world for many months. And so this was an outbreak in Guangdong province down here in the south, November 2002, with a short incubation period, very much like COVID, prodrome of fever, chills, headache, malaise, myalgia, and then dry cough, shortness of breath, and 10 to 20% of people requiring mechanical ventilation because their oxygen saturation is dropping. A Chinese doctor who treated patients in Guangdong province, he went to Hong Kong on February 21st. He stayed on the 9th floor of the Metropole Hotel. He died in the hospital the next day. He transmitted the virus to 10 people in the hotel who flew to Singapore, Vietnam, Canada, and the US before they had evident symptoms. So they were asymptomatic. They were in the incubation period and they brought it to different countries. And the infection eventually spread to 8,000 people in 29 countries with a 10% fatality rate. And I like this poster, this tourist poster, Hong Kong will take your breath away. It certainly did. So here's the epi curve, 8,000 cases beginning in 2002, ending in 2003. These are the countries where there were cases. You see the US had 29. Most of them were in China and Hong Kong and Singapore and so forth. 8,000 cases, 774 deaths. Why were we able to stop this? Because there's no pre-symptomatic transmission. But when you start to shed virus, you're really sick, and most of the time you're in the hospital, so it's easy to control that kind of a infection. So what's the origin? It turns out that human sera collected before the outbreak have no antibodies to SARS-CoV. So this is a brand new virus in humans. The earliest cases in Guangdong were found in handlers in the exotic meat market. So they sell animals, they're butchered, and they're sold to people, and some of these animals were obviously infected. And these traders had higher prevalence of antibodies than control groups. So people then went out and looked for sources of the virus. They were looking for virus in horseshoe bats, and they found some in a cave in Yunnan province about 1,000 kilometers away. And in that cave, in various bats in the cave, they found all the pieces that would be needed to make SARS-CoV by recombination. They didn't find SARS-CoV in any bat, but they found, half the genome here and half the genome there. And interestingly, so Yunnan province, where this cave was, there are no SARS cases in the province before the outbreak. They only happened in Guangdong. So the idea was, in the province, the virus, the progenitor in a bat, probably infected another animal, like a civet cat, which was then brought to the market for slaughter, and then that infected people. So bats, civet cats, and eventually into humans. And so some civet cats in the market were found to be infected with the virus. MERS coronavirus is another zoonotic infection. Our first case in 2012, a male patient died of pneumonia and renal failure. Very rapidly, the virus was recovered, identified, shown to bind to a receptor, and it was a novel bat coronavirus, not SARS-CoV. Now, this one causes sporadic infections now and then. SARS-1 spread epidemically globally, but this one does not do that. It turns out this is spread from dromedary camels, and camels in the Middle East and Africa are endemic for this virus. When camels are born, they're infected, they get a mild respiratory disease, and then it's transmitted to humans. In these areas, people value camels. They're used as pets, they're raced, they're eaten as food, and so it's very easy to transmit to people from camels, but it doesn't transmit well among people once it enters them. And so this is a perfect example of another One Health approach. You make a vaccine for camels because the disease is rare in humans. You prevent the virus from going from camels into humans. And here's a summary of some of the outbreaks. There have been, this is the original outbreak here, but there have been sporadic numbers of cases. This is 50 cases or 100 cases here, but very small numbers every month. And so far, 2,500 cases with about a 35% fatality rate. Most of the cases here are involving camel to human transmission in the Arabian Peninsula. It does camel to camel, where camels are kept in Asia and in North Africa. And a few cases, patients have traveled unknowingly infected and brought the infection to other countries. And there was one particular example of a man who went to South Korea. He had been in the Middle East. He got infected. He was fine on the plane. He got to South Korea. He got sick. They hospitalized him. They didn't know what the infection was, so they didn't do infection control, and he spread it to a number of other people in the hospital. But once they realized what it is, they do infection control, and they can stop the spread. So this is another dead-end infection. And that brings us to SARS-CoV-2, the third pandemic of the 21st century, right? We have HIV/AIDS pandemic and 2009 H1N1 virus. This begins with a cluster of cases of pneumonia in Wuhan. In December of 2019, the seafood market is the early epicenter, And the seafood market is right here. Here's the river that bisects the Yangtze River in Wuhan. And there's the seafood market, the Huanan seafood market. And these are the earliest cases. You have in red home address of cases with epidemiological link to the market and then no identified link. These are presumably people who were infected by those initial people. But you see the epicenter is at the market. It is not at the Wuhan Institute of Virology, either campus here. And so this is where the virus spilled over from animals at the market into people. We'll talk about that in some detail next time. But of course, as you know, this virus spreads very effectively asymptomatically and pre-symptomatically. We have, as of this month, 704 million global cases and 7 million deaths. So all of these coronaviruses came from nature. None of them came from a lab. We now have 4 common cold coronaviruses that circulate in humans. They all had some origin in different animals. SARS-1, bats, civet cats, humans. MERS, bats, camels, humans. There's even a disease of pigs that emerged in China in 2018, 2019 called severe, diarrhea syndrome. Severe acute diarrhea syndrome, SARS coronavirus, which occurred on pig farms in China. And that virus was spread to the pigs from bats. So this doesn't just involve people. And finally, SARS-CoV-2, all the evidence indicates that the bat is the original host. We don't know what the intermediate animal is, but there were civet cats and raccoon dogs at the meat market, and so those are candidates for that. So these viruses that are very dangerous, not SARS-CoV-2, but say Ebola viruses and Lassa viruses, you have to work on them in biosafety level 4 laboratories, which are the highest containment, and this is what you have to wear. You have to wear a suit, a sealed suit, and oxygen and air is pumped into it, and you work in very specified conditions. And the viruses that you require this kind of approach give high mortality, person-to-person transmission, and no approved vaccines or antivirals. I was able to do a video of the needle, which is the BSL-4 lab in Boston, before it opened, and so you can find that on YouTube. It's an inside look, which no one has ever done before. So how common are these jumps from animals into humans? I think, as I said, they're very common, happen a lot. We don't know because we don't monitor for them, but the ones that produce sustaining transmission are pretty rare. We don't have lots of epidemics and pandemics. Can we predict them? No, you cannot. You don't know what it takes to infect people. And we try. So the Wuhan Institute of Virology was trying to find out if there were bat coronaviruses that pose a threat to people, and they were blamed for doing that and releasing SARS-CoV-2, which of course isn't true at all. So there's an inherent danger with trying to find viruses that could be threats to humans. But we can know what's out there. We can know all the viruses that are infecting various species, and the most important species are bats, 20% of mammals, rodents, 40% of mammals, and birds, which are all over the place. So if we look in them and see what's in them, we can be prepared. We can have antivirals and vaccines ready. In fact, for SARS-CoV-2, as I'll say next time, we should have been ready. We should have had vaccines and antivirals, and it would have prevented the pandemic. But we don't have interest in doing that for reasons that we'll talk about next time. And next time, we're going to talk about 2 pandemics, HIV/AIDS and SARS-CoV-2 COVID.